Welcome to Time News. Today we will be discussing the latest developments in the longevity industry over the past week. In this episode, we will have a total of 9 news pieces to share with you. First, let's get started with 2 brief newses of David Sinclair's team. David Sinclair's team proposed a new theory called the information theory of aging, ITOA. ITOA suggests that aging is driven by the progressive loss of dynamic epigenetic information, and rejuvenation is possible through epigenetic reprogramming to restore this information. The second one is about the latest review article featuring Sinclair, explores epigenetic rejuvenation through partial reprogramming in mice and humans. It covers classical factors, chromatin remodeling, and molecular dynamics, highlighting partial applications for treating aging and age-related diseases while addressing current challenges. Both of the articles are published in Nature Aging. In General Science, a research studies the rejuvenation of cerebral microvascular function in aged mice through heterochronic parabiosis. Brief exposure to a youthful environment enhances neurovascular function in aged mice, while exposure to aged blood accelerates neurovascular aging in young mice. This suggests that circulating factors play a role in aging-related changes in the cerebral microcirculation, which is done by the University of Oklahoma. What about air pollution? Air pollution, specifically PM2.5, is linked to health issues like chest pain and high cholesterol. Surprisingly, it doesn't directly affect how long you live, but it indirectly influences life expectancy through factors like blood pressure and certain diseases. This highlights the importance of controlling air pollution for better overall health. The study is in Aging done by Chinese scientists. In Nature Metabolism, a study from Barcelona Institute of Science and Technology identifies iron accumulation as a key driver of fibrosis and senescence in mice and humans. Various injuries trigger iron buildup, persisting in senescent cells. This iron accumulation fuels oxidative stress and the senescence-associated secretory phenotype. The findings suggest iron metabolism as a potential therapeutic target for senescence-related diseases. Space travel may also accelerate aging. Researchers from the University of Liga found that cells cultured in microgravity on an ISS displayed signs of aging-related changes, such as inhibited cell growth, increased inflammation, and alterations in cellular structure. These findings suggest that the unique condition of space induces cellular state resembling aging, possibly involving the regulation of specific proteins, which is in NPJ microgravity. The next part is about reproductive aging. Deleting the gene GSS in mice leads to age-dependent fertility decline due to a specific type of cell death in the testis. While young mice compensated this effect, maintaining normal fertility, aging mice showed increased oxidative stress, leading to reduced sperm count and abnormality. The study is in Cell Death and Disease. In Nature Aging, the study found that the enzyme CD38 plays a crucial role in ovarian aging, affecting fertility and follicle reserve. Deleting CD38 in mice mitigates ovarian aging, presenting a potential therapeutic target for addressing age-related female infertility. Finally, in aging brain, astrocytes, the supporting cells in the brain, experience reduced mitochondrial function, morphological atrophy, and altered protein-lipid ratios, impacting their structural and functional roles. Interestingly, these changes don't significantly impact the activity of neurons, the main signaling cells in the brain. This helps us understand how various brain cells change with aging, which is in Nature Communications and done by Jiaxing University. And that is all. Thank you for joining us on this week's Time News. We hope you find these updates informative and inspiring, and don't forget to subscribe our channel. We'll see you in next week's broadcast, wishing you a happy and long life.